Chymotrypsin is an enzyme that cleaves after aromatic residues such as phenylalanine, tyrosine, or tryptophan. Within the active site of chymotrypsin, we see a hydrophobic pocket, an oxyanion hole, a glycine residue, and 3 different amino acids over here. Now, these 3 amino acids, aspartate 102, histidine 57, serine 195, form a catalytic triad, meaning that these 3 amino acid residues work together in the active site in order to cleave our peptide bond after the aromatic residue. Now, what do these numbers mean at the top of each of these amino acids? This number represents the location at which this amino acid occurs. So we know that when we have amino acid chains, we have lots of amino acids. So aspartate occurs at position number 102, whereas histidine occurs at position number 57 and serine at 195. Now, we also have this glycine residue, which is actually going to be stabilizing our oxyanion, which we'll see later in the steps. Now, the hydrophobic pocket is where our hydrophobic group is going to sit. So when we take a look at phenylalanine, tyrosine, and tryptophan, they all have very bulky aromatics that are hydrophobic. And these hydrophobic parts of these amino acids will reside over here. And this, in a way, keeps the polypeptide in place while it is being cleaved. Now, in the first step, we add in our substrate, which is our polypeptide, and we add it into the active site. When it enters its active site, remember we are reading the polypeptide from left to right, so we'll cleave it after the aromatic, so we will cleave it at this peptide bond between the carbon and the nitrogen. This is where we are going to cleave. Now, if we take a look over here, we can see that this histidine is attacking the hydrogen on the serine. Now we have to think, why is this happening? This is happening because when the substrate enters the active site of chymotrypsin, the enzyme chymotrypsin goes through a little bit of a conformational change in order to fit the substrate, and this is referred to as induced fit. So when induced fit occurs and the substrate binds, this compresses the bond between aspartate and histidine. Remember, we have a bond right over here between aspartate and histidine. The negatively charged oxygen is interacting with that hydrogen. So when our substrate is added in, this bond is actually compressed. So normally histidine has a pKa value of 6. Okay, that's our normal pKa value. But after this bond compresses, the pKa of histidine changes to over 12. And we know that pKa, it's the potential of dissociating, donating our hydrogens, but we also know that our environment, so the molecules or substituents that are surrounding our molecule, are going to affect the pKa value. So since this bond compresses, it affects the histidine, and now the pKa has increased to 12. So since the pKa has increased to past 12, this makes histidine a very good base now. And we know that bases, the definition of a base is that it will be a very good proton acceptor. So it will want a proton. So that is why we see histidine over here abstracting that proton from serine, because it's a very good base and it has that nitrogen has that lone pair. So it's going to abstract that hydrogen. But now this oxygen on the serine is going to gain a lone pair. Now, this lone pair is now going to create a negatively charged oxygen, which is going to nucleophilically attack this carbon over here. Now, this carbon is part of our polypeptide, which has entered the substrate. So when this oxygen nucleophilically attacks this carbon, we know that carbon can only have 4 bonds. So what's going to happen is that carbon is going to break one of its bonds, and it's going to be this one over here. So oxygen is going to gain a lone pair, okay? And we'll see that on the next slide. So here we can see that oxygen has gained a lone pair, and it's gained a negative charge. We can also see that the serine, this oxygen, remember that this oxygen just nucleophilically attacked this carbon in the previous step. We can also see that histidine now has formed a covalent bond with hydrogen. Now what's going to happen is that this is a very short-lived intermediate, okay? This is the tetrahedral intermediate, and this is very, very interesting. This is called the tetrahedral intermediate because now carbon has 4 bonds, 4 different bonds. Before it only had 3, now it has 4. So this is our first tetrahedral intermediate. And this is a very short-lived, unstable intermediate because of this oxyanion. Now, what's important to note over here is that this oxygen is actually inside that oxyanion hole we were talking about. It's positioned in this oxyanion hole so it can interact with the hydrogen of glycine and the hydrogen of serine, and they're interacting via hydrogen bonding, which is stabilizing this oxyanion, but it's still a relatively unstable. So oxygen is going to regain that double bond it had with carbon, and as it does so, carbon can only have 4 bonds, so it must lose another bond, and it's going to be this one over here. This bond over here, this structure over here is a very good leaving group, so it's going to break off. Now, as this bond breaks off, this nitrogen over here is going to gain a lone pair, and it's going to have that extra lone pair, and nitrogen normally wants to have 3 bonds, so as it's leaving, it's going to abstract the hydrogen on that histidine. So as it leaves, we can see over here, as it left, it abstracted that hydrogen on the histidine. And this is our first product. Now remember, this was originally part of our polypeptide, but now it has become our first product. And this is what we are left with. We are left with our acyl enzyme intermediate. So once again, we can note over here is that this oxygen has regained its double bond, and we are still bound to serine. Now we have to break this part of our fragment that we are left with. Now in this step, we can see that we have our acyl enzyme intermediate, and what's going to happen is that we're going to add water. Now water is a great molecule to add in mechanisms because oxygen has 2 hydrogens which can be abstracted. Now remember, this histidine, it still has that pKa value greater than 12, so it's still a really good base because the bond between the aspartate and the histidine has been compressed, and it's increasing that pKa of histidine. So the nitrogen on histidine is going to abstract that hydrogen, and oxygen over here on the water is going to gain a lone pair, and it's going to nucleophilically attack that same carbon again. Now once it does so, we see the same thing happen. This oxygen gains a lone pair, gains a negative charge. It's going to become an oxyanion within this oxyanion hole, and we'll see in the next step that this charge is going to be stabilized with the serine and glycine once again. So over here we see that the glycine and serine are stabilizing that negative charge. We can see that histidine has abstracted that hydrogen from the water, and now it has a covalent bond with hydrogen. But now what's going to happen is that, once again, this oxyanion, this is our second tetrahedral intermediate because carbon has 4 bonds. And once again, the unstable oxyanion, it's going to reform that bond that it had with carbon. And remember that carbon can only have 4 bonds, so it's going to lose a bond. So now this is going to be our good leaving group, and this bond is going to break. So when this bond breaks, this oxygen gains a lone pair, and once it gains a lone pair, it wants to abstract its hydrogen. Remember that in our first step, serine lost its hydrogen to histidine. Now it's coming back and saying, hey, I want my hydrogen back. So it's going to abstract its hydrogen back. Now, over here, we can see that serine has abstracted its hydrogen. And now we are left with this part of our original polypeptide, which is now going to break off and become our second product. Now, this oxygen is still in its oxyanion hole, and we still see the phenylalanine inside that hydrophobic pocket, which is keeping the entire molecule or the overall fragment in place. Now in the next step, we can see over here, this is the same depiction as the previous slide, where serine has abstracted its hydrogen. We're left with this part of the fragment, and now this part of the fragment is going to be released from the active site, and this is our second product. And then we are back to our original state, where we have our catalytic triad, where they're interacting with one another. Oxyanion hole is empty again, hydrophobic pocket is empty again, and we are ready to go for another round of cleavage. And that is essentially the chymotrypsin mechanism. If we understand the first half, the second half is exactly the same. We end up with two products. And remember that enzymes do not get used up or changed in a reaction. So whatever we start with, that's what we must end with.